Hi guys, so today the topic of our discussion is electron transport chain. Okay, so what is electron transport chain? This electron transport chain, which is mostly called ETC, it is present in the inner mitochondrial membrane. This consists of four complexes, okay, and these complexes are present in inner mitochondrial membrane. Now start how many complexes are present. Four complexes are present. First complex is called NADH dehydrogenase. Okay. Then the second complex is called succinate dehydrogenase. Then third complex is called cytochrome C reductase, and the fourth is cytochrome C oxidase. Okay. So I'll tell you why these are named given to these complex. So start our video. So first thing you should remember that these all things which are related to electron transport chain, these are present in inner mitochondrial membrane. Only the one thing, cytochrome C, this is not present in the inner mitochondrial membrane. It is present in inner mitochondrial space. Okay. So this the complex 1 NADH dehydrogenase, complex 2 succinate dehydrogenase, complex 3 cytochrome C reductase, and 4 cytochrome C oxidase. Okay, this complex 1 NADH dehydrogenase, okay, this get electron when NADH, we know NADH, we get this NADH from many reactions, okay, and in the TCA cycle, in the also in the glycolysis, in the degradation of fatty acid. These all are the process which give us the NADH. Many other reactions are present in our body which give NADH. Okay, and we have already studied that this NADH gives 2.5 ATP, and some books says 3 ATP. Okay, so how this give 2.5 ATP or 3 ATP, we are going to study in this lecture. So when NADH is converted into NAD positive, so this hydrogen ion is released, and we know that hydrogen ions contain one electron, so we say that electron is released when NADH is converted to NAD positive. This electron come to NADH dehydrogenase, which is the complex 1. Okay, this complex then transfer this electron to coenzyme Q. This is coenzyme Q, and also when electron come to coenzyme Q from complex 1, it is then sent with, it is then transported to cytochrome C reductase with the complex 3. So here you should know that complex 1 coenzyme Q, it is then transported to complex 3. So complex 2 is not involved when NADH is involved. Okay, so from cytochrome C reductase, it is transported to cytochrome C, electron is transported to cytochrome C. So this is the reason that electron is transported from cytochrome C reductase, or also it has another name, cytochrome BC1. So from cytochrome BC1 or complex 3, it is transported to cytochrome C, and it causes a reduction of cytochrome C, because electron when given to something and somebody, it means we are reducing that thing. Okay, so electron is transported to cytochrome C, it means it is reduced now. So the complex 3 causes a reduction of cytochrome C, that is the reason it is called cytochrome C reductase. Okay, then from cytochrome C, electron transport to cytochrome AA3 or complex 4. Okay, so cytochrome C gives electron to complex 4. So it means complex 4 cause the oxidation, because it is now gaining electron from the cytochrome C. So oxidation of cytochrome C is done by the complex 4. This is the reason we call this cytochrome C oxidase. Also another name is cytochrome AA3. Okay, so this can be asked in the question, what is another name? AA3 of 4, complex 4, and complex 3 has another name BC1. Okay, so another thing I want to tell you, that when electron are moved from complex 1 to coenzyme Q, okay, 4 hydrogen atom, how many? 4 hydrogen atom are pumped. These are pumped, not these are diffused, because electron carry the energy, and this energy is used, this energy is coupled for the pumping of hydrogen ions into the inner mitochondrial space from inner mitochondrial membrane. So hydrogen ions are transported from inner mitochondrial membrane to inner mitochondrial space when complex 1 is used. Okay, so when complex 3 is used, so electron from complex 3 to cytochrome C is moved, 4 hydrogen ion further move from mitochondrial membrane, inner mitochondrial membrane to space, inner mitochondrial space. Okay, so 4 hydrogen ion further move, and then when electron from cytochrome C come to complex 4, Okay, during this electron movement, energy of this electron is derived, and this energy is used to pump two hydrogen atom. Okay, so this thing you should remember, that four hydrogen atom in complex 1, in complex 3 also four hydrogen atom, and when electron is moving toward the complex 4, it is now, look, when electron is moving from higher to this level, its energy is used up, okay, because it is pumping hydrogen atom. So in the laws it has very less amount of energy. That's the reason only two hydrogen atom instead of four are pumped from mitochondrial membrane. Okay, you should know, from internal mitochondrial membrane, these are pumped to internal mitochondrial space. Okay, so these hydrogen are transporting. This is the NADH dehydrogenase complex 1 uses when we take NADH. But when we have FADH, okay, we have NADH and FADH. Okay, we know that NADH give 2.5 ATP and FADH give 1.5. Some books say is NADH give 3 ATP and it give 2 ATP. Okay, so when FADH is converted into very good, into FAD positive, and this gives one electron, and this electron then transport from succinate dehydrogenase with complex 2 to coenzyme Q, and then from coenzyme Q it is transported to electron transport chain. So you should know that when we use complex 2, complex 1 is not used. When we use complex 1, complex 2 is not used. Okay, and one more thing, when we use complex 1, 4 hydrogen atom are pumped, but when we use complex 2, no hydrogen atom is pumped during succinate dehydrogenase to coenzyme Q movement. That is the reason we only pumped 6 hydrogen atom when we use FADH, because 4 hydrogen and 2 hydrogen, only these are pumped. This complex does not pump any this does not pump any hydrogen atom. Okay, so totally we get 6 hydrogen atom when we use FADH, and 10 hydrogen atom, 4, 4 and 2 when we use NADH. Okay, so these hydrogen atom which are pumped, these are pumped continuously. So I should write the pump here like in this manner. So this is the pump which continuously pumping the hydrogen atoms from inner mitochondrial membrane, this is the membrane, okay, to inner mitochondrial space. So these hydrogen are accumulated in a huge amount in the inner mitochondrial space. So when these hydrogen atoms are in large amount, in large concentration, when these hydrogen ions are moved down this concentration gradient, this gradient energy is coupled with the help of complex 5. So complex 5 is ATP synthase. Some books say complex 5 is ATP synthase. So hydrogen atom are moved from this complex ATP synthase down this concentration gradient, and this concentration gradient energy is used in the formation of ATP. Okay, so this is the thing, by the formation of ATP by using hydrogen and concentration gradient. This is called chemoosmotic theory. Okay, so now I'm going to tell you how 2.5 ATP are formed during the NADH. Okay, so you should remember that 4 hydrogen atom when pumped into the from membrane to into intermitochondrial space, when these diffuse their concentration gradient. Okay, this diffusion give 1 ATP. So 4 hydrogen when used give 1 ATP. Okay, gives 1 ATP. So how many hydrogen atom are pumped when we use NADH? 4, 4 and 2. 4 is equal to 1 ATP, 4 is equal to 1 ATP, 2 ATP, and 2 is equal to half ATP. So when we use NADH, 4, 4, 2 ATP, ATP, half ATP. So total we have 10 hydrogen atom when we use NADH, and we have 2.5 ATP. But when we use FADH, only 4 these hydrogen atom and 2 these hydrogen atom are pumped. These 4 hydrogen atom are not pumped from succinate to coenzyme Q. Okay, so 4 and 2 total we pump 6 hydrogen atom, which is equal to 1.5 ATP. Okay, so this topic of electron transport chain and coupling of the electron transport chain which with the ATP is done here. If you like the videos, please like and subscribe my channel, and see you in next topic. Thank you.